Thank you for the introduction. Hello to all. I'm Eunyoung Kim from Bioinformatics Intelligence Laboratory in Jeju, Korea. First of all, thank you for all to the CAMDA and the ISCB organizers for giving this time slot for me. I'm going to present my recent research. So as he introduced, the title is DeSIDE-DDI. This is a table of contents for today's webinar. As you can see from the title, the research topic is about the DDI prediction. So I'm going to start with the introduction with what DDI is and why it is so important. So I will start from why we need to predict these drug-drug interactions. So before going to that, I want to talk about the process of drug discovery and development. Drug discovery is the process that new candidate medications are discovered. As you can see in this figure, it consists of many procedures. To develop one drug, around 5,000 to 10,000 compounds are selected as candidates. Then these candidate compounds are tested through the preclinical and clinical trials. Each step takes huge amount of time and cost. A recent study reported that the average cost of developing one drug is about $1.3 billion. So, if it is such a long and laborious process, then how many new drugs were approved previously? The figure shows the annual number of FDA approvals since 1993. You can see that the number is quite small. It means that most of the compounds actually fail to pass the discovery and development process. Of course, there are many reasons for failure in development. Among them, one of the major causes is undesirable drug effects, such as drug toxicity and the adverse drug reactions. This left figure shows how hard for compounds to pass clinical trials. As you can see, the success rate is very low, especially at phase 1 and 2. And the right figure shows the reasons for failure in phase 2 and phase 3. The second top reason of failure is drug safety. These safety issues in drug development would be drug toxicity and adverse reactions. These adverse drug reactions is a broad term for unwanted or harmful reaction after drug administration. The term, the side effects, is also used interchangeably while it indicates any effect other than the intended therapeutic effects. In this webinar, the ADRs and side effects are both used to term the undesirable effects. Unlike toxicity, detection of ADRs is done with post-market observation. There is a representative system called FAIRS. It stands for FDA Adverse Event Reporting System. It provides adverse event reports submitted by experts, consumers, or manufacturers since 1968. From this data, we can see the importance of drug side effects. The figure shows the number of reports with categories of severeness. More than half of reports are categorized into death and serious labels. As you can see in this slide, there are many side effects in marketed drugs, and among them, some might be the consequences of multiple medications. So let's move on to these taking the multiple medications, the term polypharmacy is literally taking multiple drugs at once. This is important because many diseases are complex to cure with only one drug, and in general, the risk of side effects increases with larger doses. The polypharmacy approach has the advantage in treating complex diseases such as cancer with less amount of drugs. Also, it is very important because the major subject of polypharmacy is the elderly people or the patients. Many people take some multiple medicines at once, and the number of cases increases in proportion to age. So, if these drugs are just independently react and they just have their own reaction, it not be any problem, but here comes the interactions. They actually interact each other when they come into our body. So drug-drug interactions, called as DDIs, is the change in a drug's effect when the drug is taken together with another drug. There are mainly 4 types of DDIs, but these DDIs generally mean the bad interactions. Of course, these DDIs may result in positive effects such as synergy. However, not everything has good signs. It may result in undesirable effects. Here is a famous example of a drug pair having side effects. The left compound is the warfarin, an anticoagulant, and various interactions have been reported with the. One of them is the gastrointestinal bleeding when it is taken with the NSAIDs. So, in this research, I wanted to computationally predict these DDIs. There are mainly 2 points that I wanted to handle. The predicting interactions of unknown compounds is a challenging problem, and the DDI mechanisms are not well studied. Therefore, I wanted to predict polypharmacy side effects with analysis and how and why this happened. Due to this, I used links L1000 data, which is the gene expression signatures, and applied deep neural network to construct the model. This is the overview. The model consists of 2 parts, feature generation model and DDI prediction model. Different data sets were used and processed for each model. The given drug pair and side effect data, each drug feature is obtained using the feature generation model, and a side effect is embedded to latent space. Then these drugs pass through the neural nets, having the gated linear units to consider the pair information. Finally, using these latent representations of drugs and the side effect score is calculated. The model is evaluated considering 3 DDI cases, and it was validated with an independent data set of DrugBank. So let's move on to some more details. I used different data sets for each of these 2 models. Firstly, to construct the feature generation model, I used Link's L1000 assay data. This is high throughput gene expression assay that measures the mRNA transcript abundance of 978 landmark genes from various human cell lines. It provides 5 different levels of data from raw file to the pre-processed at each step. Among them, I used the level 5 data, which means the differential gene expression signatures. Pre-processing was done to retrieve compounds with SMILES information, so the final number of the compounds that I used is 19,156. Moreover, the compound features were calculated to train the model. For the DDI prediction model, I used two-size data that contains relations between drug pairs and side effects. That were these database was pre-processed from the pairs, and the number of drug pairs varies according to each side effect. There is very common and rare side effects. I extracted the side effects with more than 500 drug pairs as a minimum. This table shows the number of data before and after preprocessing. The last data set is the DrugBank for external validation. I downloaded two versions and preprocessed as two-size drugs. This table shows the number of drugs and drug pairs for each version, and unlike two-size, DrugBank data has one binary label for one drug pair. Now let me move on to the model architectures. The feature generation model is a simple neural network with one hidden layer. As I mentioned, I used this fingerprints, this compound structural information and compound properties as features. Each feature passes separate neural nets and is concatenated to calculate the output. The purpose of this model is to generate drug-treated gene expressions for all compounds. Since only a small proportion of compounds were overlapped between links and two-size, not all compounds had expression signatures. So to utilize all drugs in two-size, I decided to generate based on the gold standard dataset. So the model was trained to fit to this given links data. On the other hand, this DDI prediction model is a little bit more complicated, consisting of two key models, this co-administration model and the score calculation model. The co-administration model is developed to express co-administration effects to gene expression signatures. Here, the GLU, gated linear units, are used to control the information flow with the consideration of two drugs. It is a simple metric of sigmoid activation function that calculate each value to range between 0 to 1. Then this is multiplied as feature weights. So this can help the model to distinguish drug pairs with the same drugs. This heat map shows how GLU affects the original features. Each row is drug feature of the same drug, but in combination with the other drug. Then, the calculated drug-latent representations and side effect embedding were put into the side effect score calculation model. Inspired by the knowledge graph embedding method, I applied translational embedding method to make two related drugs to be close in side effect spaces. So each drug of a pair is projected to a side effect space. Then the scoring function is defined as the distance between drugs on that side effect space. Since the current DDI pair information does not have the direction, so the both ways are considered. Then the loss function is defined as the margin-based loss. So with this loss function, it makes more gaps between scores of positives and scores of negatives. And this works on each side effect. So before moving on to the results, I wanted to make it clear how models were trained and validated. In this study, there are mainly 3 cases of interactions that can happen. Unseen interactions, one unseen drug, both unseen drugs. Here, unseen means that the data was not used for training. So therefore, it is new to the prediction model. This unseen drug can represent newly found compounds in the real world. Also, for each case, I split data into training and validation sets. Since the frequency of side effects varies, I applied the stratified split. Here, instead of k-fold cross-validation, I randomly sampled 1% for validation because of the large data size. Also, the process was repeated 100 times to obtain the optimal threshold to label predicted values to 0 and 1. Also, there is another point to consider, the process of external validation. As I mentioned in the previous slide, the DrugBank has DDI information, but the level of description is quite different. It describes at molecular level. The labels indicate the existence of DDIs. To handle this problem, I generated the triplets by combining drug pairs with all side effects. Then I predicted and counted the number of positive side effects for each drug pair. Here, I assume that if the number of side effects is greater than the average number of side effects in the true positive data set, that drug pair will have the interactions. So for example, if you look at this figure, you can see the predicted labels, the number of positive in this predicted label is 3. So if the threshold is 2, the drug pair of drug 1 and 2 is positive since the number of positive is 3 here. But if the threshold is 3, this drug pair is labeled as negative. Now I will show you the results. First, this shows the performance of the feature generation model. The table shows the performance in various metrics. It may seem the model is not working if you look at the correlation value, but if you look at this C-index, the model can predict the odor. And also, the bottom plots show the distribution of the gene expression data. The predicted values show higher kurtosis than the original distribution, and this might be the reasons of the relatively low correlation of this model. Then this shows the performance of the DDI prediction model. As you can see here, the constructor model showed good performance in all cases compared to the previous studies. Here I compared my model with two previous studies. One is the decagon, that was the first prediction model for polypharmacy side effect, and it showed a very remarkable performance. And it, in many recent studies, the decagon is compared as the baseline. And also, I compared with this another work by the Shankar et al. And this AM model was compared because they also utilized the Lynx L1000 data as features. Actually, in this plot, I implemented their model architectures, but of course, the model showed better performance in that paper. Though you can see from this slide, you can see that the decagon was not able to predict the interactions with new compounds, so I could not list the performance here. And the proposed model performed better compared to this AN model in case 2 and 3. And also you can see that if the drug is not trained in this model, it is quite hard to predict the interactions. Also, I looked into some more specific performance for each side effect. In this table, you can see the best and the worst performing side effects. You can see that the performance variance is not quite large. And also, I looked into the side effect specific performance, and this performance is not related to the frequency, which is quite interesting here. And this is an additional result. I extracted some billy-related disease terms because I originally presented this work at the CAMDA. I extracted some billy-related terms in here, and my model actually worked pretty well for all of these terms. Then I further validated the predicted triplets in case 1. In the case 1 is predicting the unseen interactions. The model predicted around 19,000 negative triplets as positives, but in fact, the negatives are not real negatives. So I looked into those drug pairs in DrugBank, and around 14% were found in the DrugBank database. Also, among the positively predicted triplets, I could find previously known fatal drug interactions. And as you can see in this table, like the warfarin and the related drugs and their already known side effects. The next step is the external validation with DrugBank. With the external validation strategy explained previously, I predicted all possible drug pairs in the old DrugBank published in 2016. Among predicted positives, most were false positives. Then, these predicted pairs were searched on the new version published in 2020, and the proportion of true positives increased by 35%, and this indicates that the model can capture potential interactions. And also, a permutation test was performed to validate the false positives. The same number of false positives were randomly sampled and compared how many positives were included. You can see how this p-value is low. And lastly, there are interactions of all cases mixed up in DrugBank. So I checked how many of them are in each case in terms of two signs. I identified which categories the drug pairs belong to. I found that only 13% of validated drug pairs were case 1, and the others were case 2 and 3, which are more difficult prediction tasks. These external validation results show that the proposed model can capture DDIs of all cases. So you can see more specific DDI cases in the DrugBank dataset. Not only these validated false positives, but all these drug pairs in the DrugBank has more difficult cases from the view of the two sides. You can see around more than 80% are the harder cases. Moreover, I conducted this ablation study to examine which model is essential in predicting DDIs. The baseline is the original model, and the model without feature generation model, without the co-administration model, here it says the without GLU, and the model without the score calculation model, here the green one, the without DDI model. So these three models were compared to the original model. And you can see how performance actually dropped without each model. And this results indicates that both the co-administration model and the embedding contributed the most to the model performance. Also, this co-administration model actually contributed to not only the performance increased, but also handling the mirror pairs. If you look at the figure A, you can see that the model predicted almost the same scores when the order of pair is flipped. Also, figure B and C shows that the scores are not biased to a single drug's effect. Here, the assumption is that if a polypharmacy side effect is the result of the single drug side effect, the model may predict well with the information of only one drug, but you can see the correlations are relatively low compared to this mirror pair. The last result is the feature analysis. I wanted to see how drug features change by the pair drugs. The heat map shows the changed features of this one drug, cyclophosphamide, affected by each pair drug. The hierarchical clustering was done considering the correlation of features, and the drug categories of each cluster were examined by the drug ATC codes. Expression values of several genes were changed with the co-administration, and you can see some different patterns were detected for each cluster. And it actually showed different side effect patterns. This table shows the frequent side effects of each cluster. And also, the bottom, the PCA plots show that how those clusters are far apart from each other and from the original point. And in more details, I brought this case study. Among all genes, I focused on the weighted genes. I searched on PharmGKB database for any known compound pathways. Then I found that the weighted genes are found in each pathway of the drug pair, this cyclophosphamide and salicylic acid. You can see these highlighted genes were actually found in the top 100 weighted genes. So, in summary, I constructed the deep learning-based prediction model for DDI drug-drug interactions. I used the drug-treated gene expression data to consider indirect interactions at transcriptome level and apply the graph embedding technique to successfully train the model. Also, the simple GLU was adopted to calculate feature weights and mimic the co-administration effects. The proposed model showed quite good performances, and it showed the possibility to predict interactions with novel compounds. This is the list of the references, and thank you for listening. I want to give some special thanks to my PI, Professor Hoo-jung Nam, and my lab members. Thank you. If you have any questions, feel free to ask.
Thank you. Can you hear me? Okay, I think it
Yes.
should be fine. Thanks a lot for your really nice presentation, the extended version of what was shown at CAMDA. While waiting for the questions for the attendance, I would have a few, in fact. You were showing this, I think for me it's quite intriguing that there is a different performance depending of the side effects, right? Could you go back to the slide? That the prediction differ depending what side effect you are looking at. Yeah, one more. Yeah, here.
One more
No,
here.
no. Yeah, this one.
Uh,
No,
the
one
next one.
earlier. No, this
Uh,
one. Sorry,
yeah.
not. uh,
Uh,
mm, one back
one back.
here.
Yeah.
So you have best performing side effects and the worst performing side effects, and the best performing, yeah, they're pretty high, 0.95. Could it be that the best performing side effects and the worst performing side effects, they differ in terms of the complexity of the side effect? I mean that the more
Um.
pathways needs to be is involved in the side effect, and that's why it's more difficult to predict from the expression profiles.
Oh, that is work. That is quite work to look into that. I didn't really go into why these side effects are
Yeah.
easy to predict and hard to predict. I just looked into the that the number, just the frequencies. I just
Yeah.
thought that if the frequency is quite high, the model will predict well, but
Mhm.
I confirmed that there is no correlations between the frequencies and the performance. So, oh, that is a very great
and
questions.
and
Yeah.
then maybe to follow up to this, because you are using this L1000, yeah, links database.
Yes.
So it could be that um, for the complex side effects, there might be some more, how to say, more specific things involved which are not caught properly based on the L1000, because here we are looking on less than 1000 mRNAs expression, right? And this gives a very good overview for many applications of the whole complexity of the human transcriptome, which is almost over 200,000. But maybe in some very specific um, reactions, this 1000, let's say, markers, it's not enough to capture the full characteristic of the effect.
Yeah, that is true. Yeah, only we cannot explain these complex diseases with only
Yep.
978 genes. Yeah, that is why that is one of the limitations in here, but it may be overcome if there are more experiments and more
Yep.
data. Yeah.
I mean, yes, you'd had to have, I mean, maybe experiments not only on this 1000, but in the broader for the whole transcriptome. But let's say this might be difficult in terms of the organization and also the costs, right?
Yeah. Yeah,
I'm
well, I can make um I can use other interaction data to cover up other information instead of the experiments. I
mm,
can use the PPIs or the gene-gene interactions data to cover those um, more informations from the other genes.
mhm.
Yeah. Um,
um, I think I'm not sure that I fully understood this gating mechanism where you taking where there will be an interaction between drugs. So how in fact it's working? I mean, I'm not sure that I understand. So you have the expressions from the both, right?
yes. Here, can you see the point?
Yeah, I see. I can see it.
Yeah. These are the expressions of each drug,
Mm-hmm.
and I just concatenated. These are just feature vector, and I concatenate these two vectors. And with this gated linear unit, it's just the um, simple, just sigmoid functions.
Mm-hmm.
If you put this in here, this function actually outputs these values to uh, between 0 to 1. So
Mm-hmm.
this value will be 0 to 1. And here, actually, I reduce the dimension to be equal to the gene expression data. So the dimension is 978 and it doubled up and it actually reduced to the original dimension. So this is like a weight, feature weight. So um, I element wise I applied element wise multiplication to this original expression. So with this feature vector, um, these expressions were changed. So while the model is training, it will automatically learn which genes are important by these sigmoid functions. It
Mm-hmm.
will determine which genes will should be selected.